Now that we have an understanding of the general structure of viruses, let's take a look at the modes of viral multiplication. As we've already discovered, the host cell is required for viral multiplication. And there's a specific interaction between the host cell and the virus. After the virus has made its way into the cell, it takes over the machinery of that cell in order to replicate many copies of itself and release itself out into the environment. The multiplication cycles in animal viruses involve these stages. Adsorption, which is attachment to the host cell in a very specific manner. Penetration, or entry of the viral genome into the host cell. Uncoating, or release of the viral genome from its packaging. And synthesis of the new viral products. Assembly of new viruses, and then release of those viruses out into the surrounding environment. Let's begin by taking a closer look at adsorption. Adsorption is sticking to the surface of. So the virus is going to encounter a susceptible host cell. The virus essentially sticks to specific receptor sites on the cell membrane. The membrane receptors are usually things like glycoproteins that the cell requires for its normal functioning. For example, the rabies virus affixes to acetylcholine receptors of nerve cells in mammals. Because of the exact fit that's required, viruses have a limited host range. This limited host range can either be restricted, intermediate, or broad. Restricted would be an example, say hepatitis B. It infects only the liver cells of humans. An intermediate host range is exhibited by the poliovirus. The poliovirus infects intestinal and nerve cells of primates, so humans, apes, and monkeys. And a more broad host range would be exhibited by rabies, because rabies can infect various cells in all mammals. Remember, there were two forms, either enveloped viruses or naked nucleocapsids. The enveloped form uses glycoprotein spikes, as you see here, that attach to the specific receptors in the host cell membrane. Naked nucleocapsids use molecules that are attached to the capsid itself. Once the virus has adsorbed to the cell membrane, it needs to penetrate and then become uncoated. The flexible cell membrane of the host is penetrated by the whole virus or its nucleic acid, where the capsid stays on the outside. We can see endocytosis, where the entire virus is engulfed by the cell. In this image here, we can see the specific attachment that the virus is being fully engulfed by the host cell. After it's fully engulfed, it's contained in a vacuole or a vesicle, and enzymes within the vacuole will dissolve the envelope and the capsid. At this point, the virus is uncoated, and we have free DNA within the cell. Another mechanism for this is what we see when the viral envelope fuses directly with the host cell membrane. Here, the viral envelope makes a specific attachment and fuses with the host cell membrane, releasing the viral contents directly into the cell. After the virus has penetrated the cell and become uncoated, it's time for the free viral nucleic acids to exert control over the host's synthetic and metabolic machinery in the phase called synthesis. In DNA viruses, the virus enters the host cell's nucleus, where they're replicated and reassembled, as exhibited over here on the left side. There's a complex sequence of events that goes on, but basically the DNA is in the nucleus and it's transcribed in two phases. So in the early phase, the viral DNA codes for all the enzymes that are necessary for replication of its own DNA in order to allow it to make many more viral capsids. In the later phase, the viral DNA that codes for the structure of the proteins involved in the capsid shell is transcribed. Sometimes this DNA will remain free in the nucleus, and other times, in double-stranded DNA viruses, the DNA can be inserted into the host DNA. RNA viruses, in contrast, replicate and assemble themselves out in the cytoplasm. And this makes sense because the RNA in many RNA viruses is essentially messenger RNA, so it can go right to ribosomes and produce proteins directly. In this slide, we can see a variety of modes of synthesis in viruses. The top one exhibits the common DNA genome. If we have a DNA strand, then that DNA can be replicated and we can make RNA from the DNA and then eventually end up with a protein. However, when we look at RNA genomes, things are a little bit more complex. As you recall, we can have positive RNA genome. We can have a negative RNA genome that's in the virus. In the positive sense, that RNA genome can be directly translated into proteins, and more RNA can be made, which can then be packaged into capsids for release from the cell. In the negative strand situation, we need to make a positive strand by copying the negative strand, and from that we can then make the proteins and more negative RNA to package into the new viral capsids. So this should give you an idea of the diversity of various viruses and how they might go about taking over the cell's machinery. Now let's look at assembly and release. Mature virus particles are constructed from this growing pool of parts because they've taken over the machinery of the cell, and now that cell is manufacturing viral nucleic acids as well as viral proteins for the capsid. Non-enveloped and complex viruses are released when the cell lyses or ruptures. However, enveloped viruses need to be liberated by budding or exocytosis. You can see that process down here at the bottom of the screen, in which a piece of viral DNA is pushed out through the membrane into a bud and then released from the cell. The micrograph over here on the right is of HIV leaving host T cells. Let's take a look at a quick animation that shows the release of these.
Enveloped viruses are usually released from the host cell by a budding mechanism. First, viral spike proteins are inserted into the host cell membrane. Next, the inside of the host cell membrane becomes coated with viral matrix protein. The viral capsid then becomes completely enclosed by the region of the cell membrane into which the spikes and matrix protein are embedded, and the virus is released by budding.
So what happens to the host cell? Obviously, if they have to lyse or burst in order for the new viruses to get out, then those cells are destroyed. So the fate of most cells that are infected is death. If the cell does live, there can be some cytopathic effects. For example, virus-induced damage to the cell that alters its microscopic appearance. Cells can become disoriented, change shape or size, or have intracellular changes. For example, in human epithelial cells that have been infected by the herpes virus, you'll see multinucleate giant cells. We'll also notice that there is no junction between the cells. They're no longer in a side-by-side arrangement because the virus has disrupted these cohesive junctions. In the figure on the right, we'll see an illuminated image of human cells that are infected by the cytomegalovirus. You'll notice these large green inclusion bodies. Again, there's disruption between the cell junctions, so these cells are fairly well disassociated, as in they're not aligned side by side. So inclusion bodies are compact masses of viruses or damaged cell organelles that are trapped inside the cell. The fusion of cells results in syncytia, or multinucleate cells. Now, sometimes we'll see persistent or latent infections. This occurs when some cells maintain a carrier relationship with the host cell, so they don't burst or lyse right away. Persistent infections can last for a few weeks or the entire life of the host. This is where the virus will remain in the cytoplasm or incorporate its DNA into the host's DNA. When the DNA becomes incorporated, we call this a provirus. Sometimes we see a chronic latent state, where the virus remains permanently in such things as cold sores or genital herpes or chickenpox or shingles. All of these are herpes viruses, and they'll remain in a chronic latent state, and you can have multiple outbreaks. Sometimes they'll go into quiescence. Often it's stress that causes them to be released from the nerve cells, and then we'll see another outbreak. And some viruses actually cause cancer. They enter the host cell and permanently alter its genetic material. These viruses are called oncoviruses or oncogenic viruses. The effect on the cell is called transformation because they've been transformed into cancer cells. It's suggested that upwards of 20% of human cancers are caused by viruses, and that's just the ones we've identified so far because this is a relatively new idea. Recently, we've heard a lot about genital warts and cervical cancer. We also know that hepatitis B causes liver cancer. One of my colleagues works on the polio virus vaccine and its relationship to mesothelioma. There's a really interesting book that's been written about his research called The Virus and the Vaccine, where we deal with all the controversies of vaccinations, especially when some of these vaccinations that were ordered by the government are now being found to have caused cancers. Really interesting read. Viruses that infect bacteria have a slightly different mechanism of infection. These viruses are called bacteriophages. They're the complex phages we've looked at earlier. Phage is the Greek word for eating. Most bacteriophages contain double-stranded DNA, but they can run the whole spectrum. They could have single-stranded DNA or RNA nucleic acids. They're of particular interest to microbiologists because often the bacteria that they infect become more pathogenic for humans after they're infected. The most widely studied of the bacteriophages are the T-even phases, such as T2 and T4. They have an icosahedral capsid head, a central tube, as well as a collar and a base plate that has tail pins and tail fibers. The bacteriophage attaches to the cell but does not enter the host cell. It simply injects its viral nucleic acid into the cell. That viral nucleic acid will then take over the cell's machinery and produce more bacteriophages, which will eventually be released from the cell. Bacteriophages go through similar stages as animal viruses. They're adsorbed, but they do not enter the cell because they're injecting their genetic material into the cell. Entry of the nucleic acid into the host cell causes cessation of that cell's DNA replication and protein synthesis. The host cell's machinery is essentially hijacked for viral replication and synthesis of the viral proteins so that more capsids can be produced and released from the cell. As the cell produces new parts, they spontaneously assemble into the capsid. Eventually, the cell lyses and releases all those new phages into the environment in the surround. The cell here has ruptured and released numerous virons that will now attack cells in the surrounding area. There's an additional component to bacteriophage infection. They can be in either a silent phase or a lytic phase. The silent phase is called lysogenic phase or lysogeny. In this phase, there are temperate phages. These temperate are special DNA phages that undergo adsorption and penetration, but they're not replicated or released immediately. Instead, the viral DNA has been incorporated into the bacterial genome, and so it enters the inactive prophage stage. So what we have here is the DNA has entered the cell and it's now incorporated into the genome and it's hiding there. During this phase, the host cell's progeny or offspring are also going to have this same piece of viral genome. In this prophage stage, the bacteria is said to have undergone a lysogenic conversion. That is, it has acquired new traits from this temperate phage. At some point, though, the cell will exit the lysogenic stage, in which the virus is essentially dormant, and enter a lytic stage. You can see over here on the left an illustration of the lysogenic stage, where the viral DNA becomes latent as a prophage. That viral DNA can be incorporated into the viral genome, and it can be duplicated along with the genome of the cell as that cell divides. At some point, though, the host cell will leave the lysogenic stage and enter the lytic cycle. In the lytic cycle, phages will inject their viral DNA into the bacterial cell. That viral genome will be duplicated, which means replication of the virus's genetic material. A whole new host of bacteriophages will be produced. They're put together from all the parts in here. As they mature, they will then lyse or burst out of the cell, scattering thousands more bacteriophages into the local environment. So again, a few differences. We don't have to have the penetration and uncoating phase. And we can go into a lysogenic or hidden stage. And a bacteriophage can go into a lysogenic or hiding phase inside the host organism. Here's a quick summary table of bacteriophage versus animal virus multiplication. You might take a look at this and clarify some of the differences. So before we finish up this section, let's have a concept check. Corynebacterium diphtheriae becomes more pathogenic when a bacteriophage called beta is incorporated into its genome. What is this process called? Is it transposition, transformation, lysogenic conversion, or lytic cycle? Well, transposition isn't something we've dealt with. Transformation is when a virus causes a cell to become a cancer cell. The lytic cycle is when bacteriophages are lysing or bursting out of the host cell. And the lysogenic conversion is indeed when the bacterium has been converted to a more toxic form by the incorporated viral genes. How about another one while we're here? How are the infectious cycles of viruses different in bacterial and eukaryotic host cells? The virus genome does not enter a eukaryote. The virus genome does not enter a bacterium. The capsid does not enter a eukaryote. And the capsid does not enter a bacterium. Well, in order to have any effect, the genome has to be entered into the cells, so we can cross both of those out. But we did note that in eukaryotes, the capsid can be drawn in by endocytosis, so that one can get crossed off. We did learn now that the capsid does not enter the bacterium, so that would be the correct answer.